Ron was the senior toxicologist at the National Toxicology Program for more than almost 30 years. And we worked together at the very beginning when we were both a lot younger, and on setting up studies, and he was involved in many, many bioassays. This was probably the largest. It is one of, if not the largest one ever done at the NTP. And you can forward this way, okay, or you can use this, right? So Linda Birnbaum has given us this. The first thing she wanted me to say here is that this is a final report. It has been peer reviewed internally and externally, and the results are not going to change. Right. Ron?
Thank you. Thank you, Deborah, and good evening, everybody. I'll be talking about the partial results of the NTP carcinogenicity studies on cell phone radio frequency radiation. So the title's right, but I'm not Linda Birnbaum. She's the director, and I'm retired 8 years from the National Toxicology Program at the National Institute of Environmental Health Sciences. So just for people who are not familiar with the NTP, and as you see in the government we use a lot of acronyms, so I may get a few wrong myself. This was an interagency program created in 1978. It's headquartered at the National Institute of Environmental Health Sciences in Research Triangle Park, North Carolina. And it's composed of agencies within the Department of Health and Human Services, the National Institutes of Health, CDC, Center for Disease Control, and the Food and Drug Administration. And the purpose of this is that the agency, the National Toxicology Program, does research to evaluate thousands of chemicals in toxicology studies, and the results are then communicated through technical reports, scientific publications. The NTP also has other activities. The report on carcinogens is one which describes whether environmental agents are reasonably known or known human carcinogens. There's other activities going on within the NTP, the Office of Health Assessment and Translation. This is more translating technical information into understanding of human health, and the Agency for Evaluation of Alternative Toxicological Methods. This is one in which it's an attempt to move away from using animals to the extent that's possible. So the mission and goals of the National Toxicology Program are to evaluate agents of public health concern by applying tools of modern toxicology and molecular biology. So it's a combination of animal studies and mechanistic studies. The NTP coordinates then testing programs within DHHS. Prior to its formation, there were toxicity studies conducted separately at each of the agencies, and this was a way of bringing them all together under one program. Also, developing methods to reduce, refine, or replace the use of animals, and there's more of an attempt now to make use of in vitro studies to understand mechanisms by which agents may be causing adverse health effects. And then to generate the data to strengthen knowledge about potential hazardous agents and use this information for assessing human risk. And it's then communicated to health regulatory agencies, so agencies like the United States Environmental Protection Agency, OSHA for Occupational Safety and Health Administration, use this information for determining safe levels or levels that need to be reduced to protect workers and the general public. Okay. Okay. Just one comment through this in terms of the value of rodent toxicology studies. We use rodents to understand events that might be occurring in humans. And the reason for using rodents is because these are show the same types of processes leading to disease between animals and humans. So they become a model for human health effects. The 2-year exposure covers a large part of the lifespan of the rodent, and reasons that they are used are because every known agent which causes cancer in humans, has been shown, when adequately tested, to cause cancer in rodents. And about a third of the human carcinogens were first identified in rodents. A couple of other reasons for using rodents is that in a toxicology study, we have exquisite control of exposure. So we can avoid the complications from a human study where there may be confounding from other types of exposures. And most importantly, probably, is that within the rodents, we can get an answer fairly quick rather than waiting for a disease like cancer that has a long latency before implementing safety strategies to protect people. This is a prevention approach to disease. Okay. The RF radiofrequency radiation studies were nominated to the NTP by the Federal Food and Drug Administration. There were concerns at that time for cell phone RF exposure to the head, and there had been reports in the literature that exposure to RF was associated with brain cancers in humans. Some of the epi studies show potential increases in glial cell tumors in the brain and schwannomas. This is a word to keep in mind. These were acoustic neuromas. It's part of the sheath around the nerve in the ear that may be associated with cell phone use. However, there have been inconsistencies in results, confounding factors, and biases have also been considered in evaluating whether this is a human carcinogen or not. Some of the biases, for example, are that perhaps cell phone users, when they report their use, report it more on the side of the head in which they hold their phone. Studies in laboratory animals at the time that this was nominated had not been associated with exposure to RF for tumors at other sites. However, when we looked at that at the time, we were concerned that there were inadequacies and limitations in a number of these studies. So we set out to design a study that could challenge the hypothesis that there is no effect from cell phone radiation on tumor induction in animals. And at that time, we also learned that there were studies being conducted that were associated with the World Health Organization and the cell phone industry. So we went to Geneva to hear what they were planning. And the exposures that they were planning at that time would involve animals kept in tubes and exposed for 2 hours a day. We thought that would not be an adequate challenge, so we worked with people from different agencies. One was the National Institute of Standards and Technology, and a laboratory in Zurich, Information Technology and Society, to come up with a different approach. These are reverberation chambers, which is essentially a sealed room. It's stainless steel room. It has an antenna in there, and there are paddles in this region which distribute the electromagnetic fields so that it is constant, statistically constant over time. So then this picture here shows what that room looks like when there are two sets of cage racks in that particular room. So under this condition, why this was selected was we didn't have to put animals in tubes. We could expose them for much longer time, because what we're interested in is how do you challenge to cell phone radio frequency radiation without heating animals. We know that if you expose them to too high exposure, you will create heat. If we limit the exposure intensity, we may not be understanding the possibility that there is an effect, because in these studies and in most toxicology studies, we're using 50 to 100 animals per group. However, if we cannot distinguish a risk of 1% in a study of this type, we need at least 5%. In order to see 5% in the human population would be an epidemic. So in toxicology studies, what you do is you increase the exposure or dose to the animals to see if there is an effect, and in the risk assessment, you extrapolate through down to exposures where the risk may be 1 in 100,000 with lifetime exposure. You can't get that from an animal study. So in order to challenge, what we did was we extended the time of exposure, and that's why the experiments were conducted over 18-hour periods. The other 6 hours a day was for animal hygiene, weighing animals, etc. And we had a 10-minute on, 10-minute off cycling. So one chamber would be exposed for 10 minutes with the same amplifiers. Another chamber would be off, then it would reverse. So there were 3 phases to the toxicology studies. One was in the rats, we conducted a 5-day thermal pilot study. Wanted to get a sense of could the animals maintain their body temperature at various exposure intensities. We went up, SAR is a measure of dosimetry. It's the specific absorption rate. We varied from 4 to 12 watts per kilogram. We then conducted a 28-day study to see if animals could tolerate what we considered to be a minimally thermal effect on animals, and then the 2-year carcinogenicity studies. As I mentioned, these were 9 hours of exposure per day, and we used 2 modulations, CDMA and GSM. Okay. This slide just shows the effect on temperature and why we used 6 watt per kilogram, because it was less than a 1 degree rise in centigrade in body temperature. In the 2-year studies, body weights and birth were reduced throughout lactation. In general, survival was greater in the GSM and CDMA exposed groups, but the incidence of schwannoma, which are tumors, in this case in the heart of male rats, were significant dose-response trends, as well as a pairwise comparison between the 6 watt per kilogram dose and controls. There was also significant trends for increased gliomas of the brain of rats to CDMA-modulated radiofrequency radiation, and the incidences then of heart and brain lesions were not significant in female rats, although there were some, and not in the controls. So the conclusions from the NTP study were that the combination, that hyperplastic lesions as well as tumors, hyperplasias were also seen in dosed animals. These are preneoplastic lesions, were increased in the heart and brain of male rats, and these are considered likely the result of whole body exposure to GSM or CDMA modulated radiofrequency radiation. It appeared to be higher between RF exposure and neoplastic lesions in the heart compared to the brain. In female rats, the effects were not significant for GSM or CDMA. These are partial findings in that it's not the complete report. The complete reports are expected to be public at the end of 2017 and a peer review between 2017 or 2018. Just another aspect of the studies that were also important is that within these studies, rats and mice were sacrificed at 19 weeks of age and evaluated for genetic effects. The COMET assay is one which detects DNA strand breaks, and in summary, the COMET assay identified DNA strand breaks in the brain with CDMA male rats, with GSM male rats in the cerebellum, hippocampus. Not as much in the female rat, but there was also DNA strand breaks identified in the frontal cortex of mice, both males and females. So within the U.S. government, it's the Federal Communications Commission which requires exposures to meet their guidelines. Their guidelines, I believe as Deborah was talking, were established in 1996 to avoid thermal effects from acute exposure. And they're not protective against chronic effects with even lower exposure levels. The FDA has a jurisdiction for health-related effects, but they do not require cell phone makers, industry, to provide data. Other agencies that are interested in these kinds of results are the U.S. EPA, the National Cancer Institute, and the Occupational Safety and Health Administration. Next steps, we may wait on. Okay. So I just want to mention the collaborators on this work. This was designed within the NIEHS. The laboratory ITRI in Chicago is where the studies were conducted. The engineers and physicists from the National Institute of Standards and Technology in Boulder, Colorado helped us with the design. And at ITIS is where the chambers were validated, the exposure dosimetry was worked out. They built the chambers and they were shipped to Chicago for the conduct of this work. So this is a picture of the National Institute of Environmental Health Sciences located in Research Triangle Park.